membrane-bound transcription factor site-1 protease, or site-1 protease, S1P, for short, also known as subtilisin, kexin-isozyme 1, SKI1, is an enzyme, EC 3.4.21.112, that in humans is encoded by the MBTPS1 gene. S1P cleaves the endoplasmic reticulum loop of sterol regulatory element binding protein, SREBP. Transcription factors. Function. This gene encodes a member of the subtilisin-like proprotein convertase family, which includes proteases that process protein and peptide precursors trafficking through regulated or constitutive branches of the secretory pathway. The encoded protein undergoes an initial autocatalytic processing event in the endoplasmic reticulum, to generate a heterodimer which exits the and sorts to the cis-medial Golgi, where a second autocatalytic event takes place and the catalytic activity is acquired. It encodes a type 1 membrane-bound protease which is ubiquitously expressed and regulates cholesterol lipid homeostasis via cleavage of substrates at non-basic residues. Clinical significance. Mutations in this gene may be associated with lysosomal dysfunction. See also membrane-bound transcription factor site-2 protease, site-1 protease at the U.S. National Library of Medicine. Medical subject headings. MESH. This article incorporates text from the United States National Library of Medicine, which is in the public domain.